Hey guys, it's Mr. Kennedy back with yet another video, and we're going to be talking about prokaryotes, and this is basically where metabolic diversity began. Now, whenever we look at prokaryotes, we got to remember that they come from two domains: the domain bacteria and the domain archaea. So they are, or they originated from monarans, the kingdom Monera, and they are all single-celled, and they do not have a nucleus. Now, bacteria, if we think about it, they are everywhere. They are on animals and plants, they're in animals and plants, the soil, the oceans, they're in hot environments, cold environments, you name it, bacteria are there. Now, when we talk about how the different shapes of bacteria there's three basic shapes: rods, spheres, and spirals, which are effectively called, rods are called bacillus, spheres are called cocci, and spirals are called helical. And here's some examples down here at the bottom. Like bacillus is an example of a rod-shaped bacterium. But Bordetella is an example of some sphere-shaped bacteria, and spirulina is an example of spirilla bacteria. This is like your blue-green algae. We talked about prokaryotes. We talked about their being unicellular in their shape, but their size, they're only about 1/10 the size of a eukaryotic cell. They're less than 1 micron, so they're very, very small. Their internal structure is a little bit different than eukaryotic cells as well. They have no membrane-bound organelles. And the way I remember this is, I know that eukaryotes have a nucleus, and a nucleus has a membrane around it. Prokaryotes do not, so they have no membrane-bound organelles. And they do have, I should say, ribosomes, and this is the one characteristic that prokaryote and eukaryotic cells do share. They also have circular DNA, and their DNA is naked. It's not packed around any type of proteins or anything like that. Now, when we look at variations within bacteria cells themselves, cyanobacteria, which is bacteria that is photosynthetic, its internal compartments look very similar to chloroplast. It even has thylakoids. And then you have aerobic bacteria, and aerobic bacteria, their internal compartments look very similar to the mitochondria. So they're involved in cellular respiration. And you can see both of these over here. You can see how in the mitochondria, you can see how these folded membranes here are, and you can see these same folded membranes in this aerobic bacteria down here. In this chloroplast, you can see the stacks of thylakoids. You can see them on the outside here within this cell. So they have similarities to organelles we have in eukaryotic cells. Prokaryotic cells can be divided by gram-positive and gram-negative staining a lot of times, and this is referred to basically to their cell wall composition. A bacteria that can be stained real easily, a gram-positive bacteria, is going to have a very simple cell wall. It's going to actually have a peptidyl glycogen cell wall. This one here is going to be your gram-positive bacteria. And this is basically made of polysaccharides and amino acids. Your gram-negative stain don't stain very well in the stain, and they are going to have a more complex structure, and they're going to be this one, which is your lipopolysaccharide, and that is your gram-negative stain. That's just lipids and polysaccharides. So just realize that the positive and negative deal with the complexity of the cell wall. The gram-positive is not as complex as the gram-negative. Now, when we talk about prokaryotes, most of them are motile. That means most of them move, and they have some kind of taxis, which a taxis is simply a movement away or towards a stimulus. For example, it would be moving toward food or water and away from maybe light or toxins or whatever. And the way they can move, there's three basic ways they can move. Flagellum, which is a whip-like structure behind the bacterium. Helical shape prokaryotes actually are spiral, and they move in like an undulating movement. Spirocytes are an example. And slime. Slime are like gliding mold or gliding bacteria in which they, not mold, but bacteria in which they put down a slime patch and they move across that slime patch. And last thing, or I should say yet another thing, is form and function of prokaryotes. They have what's called a nucleoid region. This is a nucleoid region. This is where their genetic material is housed. And they have plasmids, which are circular, which is their DNA molecules. They can reproduce by asexual reproduction, usually through binary fission. That simply means they split. This is not mitosis, by the way. They can also reproduce by sexual reproduction, which is not meiosis, but they do it three ways. Either transformation, where they can take up genes from their surroundings; conjugation, where they can actually form a bridge between two bacteria and exchange genetic material; or transduction, in which a virus can introduce new genes into the bacteria. And one other unique thing about prokaryotes is that they can have what's called endospores. Endospores are resistant to harsh conditions, and they can lay around for a very, very long time, 250 million years or more, until the conditions become just right for them to come out. Now, when we talk about the genetic variation in bacteria, bacteria can reproduce every 20 minutes. About 1 in every 200 bacteria that are made have some type of mutations. That's a lot of mutations. Most of the cells of your body are not your own body. Probably only about 10% are. The rest of them are these prokaryotic cells that either live in your gut or on your skin. So there's a lot of different cells that are not yours that are found within your body. When we talk about the variation within bacteria, you got to talk about how they swap genes. They do it through plasmids, which are these circular circles of DNA, which are like right down here, right? Or they can do it through conjugation, which is over here, which they build a bridge, and genetic material from this one will be transmitted to that one, or vice versa. We look at prokaryotes. We got several different types. We got photoautotrophs, which are photosynthetic bacteria. They're like cyanobacteria. You got chemoautotrophs, which actually make their own food from chemicals. Those are the ones that may live on hydrothermal vents. You got photoheterotrophs. They use light to generate ATPs, but they get their carbon from actually eating other organisms. And you got chemoheterotrophs, which they got organic molecules where they get their energy, but then they also may get their carbon from eating different ones. Then we have the ones that have an oxygen relationship. You have obligate aerobes, which means they need oxygen in order to be able to survive and do respiration. You have facultative anaerobes, which means if there is oxygen present, then they can use it, but they don't need it necessarily to survive. And then you have obligate anaerobes, where oxygen is actually poisonous to them, and they need to have an environment where it doesn't have oxygen in order to be able to survive. Now, when we talk about bacteria, bacteria can be a pathogen. A pathogen is anything that causes a disease, and some common diseases caused by pathogen or bacteria is like tooth decay, leprosy, gonorrhea, your STD, some STDs, typhoid fever, cholera, TB, etc. Bacteria are basically opportunistic. That means they normally reside in the host and don't cause any problems, but whenever the host becomes weakened, that's when they attack. So they're very opportunistic. Koch's postulate is how we use to confirm the bacteria causes disease or not. Koch's postulate is basically this. You take an organism that dies from what you think may be the bacteria. You isolate the bacteria and you grow that bacteria from that dead organism. You inject that bacteria into a healthy host, and then you see if it's exactly like it caused it to die, and you see if that bacteria that results is exactly like the one originally. If so, you figured out what bacteria or what was the cause of the death. That's Koch's postulate in a nutshell. Then also in bacteria, when they're pathogen, you have either exotoxins or endotoxins. Exotoxins are usually when a bacteria releases a toxin that's like cholera, which causes you to have severe diarrhea or dehydration. It actually releases a toxin from its body, which causes you to be sick. Endotoxins are basically found in the cell membranes of the bacteria, and they usually only release when the bacteria dies, like in salmonella or food poisoning. Now, bacteria are very beneficial, and without them, we probably would not survive. We depend on them to be decomposers, which means they break down dead organisms and return the carbon back to the soil and other nutrients. We rely on them for nitrogen fixation, which they take nitrogen from the air and make it into a form that plants can use by their nodules on their roots, like the picture over here on the right. They can help in digestion by having E. coli. They help break down cellulose in herbivores. They help make vitamin K and B12 in humans, so we need bacteria. And you may hear sometimes whenever you have a sickness and you're taking antibiotics, antibiotics affect bacteria. The doctor may tell you to eat yogurt, which would actually replenish that healthy bacteria that you have in your gut normally. And then we can use them to produce medicines like penicillin through GMOs, that's human insulin down here, or through making yogurt. This is an example of what prokaryotes are, kind of in a nutshell, and I hope that helps you.